Okay, hello everyone. Thanks for joining today the Israel-Korea Bio-Convergence webinar. My name is Yeongsun in IETO Korea. Today, the webinar is going to be started by the opening remarks by the Yaniv Goldberg in head of IETO Korea. Then there's going to be the company pitch by the 6 of our participant company today, and 3 comes from the Israel side, and the other 3 is the Korean side. There's going to be the technological explanation of cutting-edge technology. Then there's a panel discussion at the end of the company pitch for around 20 minutes. And the Ella Herrer, the IIA, are going to have a moderator. Then the CEO of KORIL, the Seongryong Kang, and then the Senior Director of Business Development of IIA, the Karina Rubinstein, having the discussion for the Bio-Convergence program. So let's start by the opening remarks by the Yaniv Goldberg, the head of IETO Korea. Yaniv, the floor is yours.
Yes, thank you, June. Thank you very much. So hello, everyone. 안녕하세요. I'm not sure if you can actually see me, but can you? Okay, maybe you can. Yes. So my name is Yaniv Goldberg. I'm the head of Israel Economic and Trade Office here at the Israeli Embassy in Korea. Our office, on behalf of Israeli Ministry of Economy, is in charge on promoting Israeli exports of goods, services, and mainly technologies to Korea, as well as attracting investments from Korea to Israel. I need to apologize in advance, since I'm not able to stay longer due to some urgent issues I need to take care of, so my apologies. This event today, with a selected group of most innovative Israeli and Korean bioconvergence companies, is another milestone of our joint effort, together with Korea-Israel Joint R&D Fund, in short, KORIL, the Israeli Innovation Authority, and our embassy, of course, to bring Israeli and Korean companies together to explore the fascinating field of bioconvergence. The field of bioconvergence, as you all know very well, has an endless potential and brings together 2 of the most scientific areas where Israel and Korea are actually taking a leading global position in past years: IT, engineering, data science, and healthcare. So much more than only 2 areas. Especially today, when new variants of COVID-19 emerge with a necessity to accelerate the process of drug and vaccine discovery, the emphasis on international cooperation, joint funding, and R&D is even greater. Israel and Korea, as you probably already know, are leading the world in terms of expenditure on R&D out of our total GDP. Each is spending around 4.5% together. With the future increased fund of KORIL of $8 million in total, more opportunities for bioconvergence projects are expected. I encourage all of you to take this opportunity to engage directly with each other and ask for a follow-up one-on-one meeting with the companies presenting today and after this webinar. This is also the place to say warm thank yous to all the participants and all the Israeli and Korean companies that joined us today. I had the pleasure of hosting AlphaTAU, for example, recently here in Korea, and I definitely hope to be able to meet with all of you whenever possible here in Seoul. Also, many thank yous to the Israeli Innovation Authority, to Karina and to Ella for the wonderful cooperation together and for your efforts towards the Korean market. Special thank you to KORIL management here in Korea. I would like to warmly congratulate Mr. Kang for starting his position as Chief Executive of KORIL here in Korea, and to thank him and his team for our partnership and also friendship. Finally, last but not least, the wonderful Mr. Yeongsun, who put a lot of energy and dedication in facilitating this webinar. So thank you also, June. I wish you enjoy this event today and learn a lot about Israeli and Korean companies doing bioconvergence together. And thank you all. Kamsahamnida. Thank you.
Thanks. Okay. Good morning, everyone. It's my honor to be invited to this webinar and to be the first company to pitch. I'll share screen immediately. Okay, I hope you can see my screen. Targetgene is an Israeli startup developing next generation immune cell therapies. Bit about the cell therapy market. It's a huge growing market today, expected to reach about $50 billion in about 6 years. And just in the Corona year of 2020, investment in cell therapies has been the highest ever and 50% more than any other year before. What Targetgene is bringing to this field are novel solutions for unsolved problems in both autologous and allogeneic cell therapies. In a nutshell, we are developing in two main directions. The first is our orphan diseases treatable by modification of blood stem cells, which will be turned into gene knockout neutrophils. And the second is a cancer, where we'll be making changes to T cells, NK cells, and so on, putting ourselves into the CAR T or the tumor infiltrating lymphocyte part of cell therapy. Of course, Targetgene also has the possibility to go into iPSCs and other avenues as well, but those are today the lead indications that Targetgene is going in. So how does this connect? First of all, cells integrate information from internal and external signals and turn on and turn off genes. And this can be seen like a very complex electronic circuit with many different logic gates. And immune cells specifically receive information and act on cells adjacent to these immune cells, either by killing or by becoming exhausted or other signals which they receive. So this can be seen as a kind of a extracellular biocircuit. If we look first at the internal circuit, here we're talking about the external circuit. And when you make changes to gene expression on the inside of a cell, you can influence how this cell acts on its environment and how the environment acts on it. One of the big problems, for example, in CAR T is exhaustion. And there are many genes which mediate exhaustion directly or indirectly, some which are just markers of exhaustion and some which actually make changes to this. In the exhausted cells, T cells inhabit the tumor, for example, but they don't do anything. And this is a problem because obviously when you treat someone, you want these cells to work, to be effective, but not to attack self at the same time. So there's many different things that need to be done to these cells, and these changes can be done with gene editing. Gene editing is done using Targetgene's TG platform. Many people, I'm sure, have heard of CRISPR. The TG system is similar in many ways to CRISPR, but has advantages over this technology. The TG system is far more specific than CRISPR, keeping the same versatility and ease of use and nearing the efficiency of CRISPR. Targetgene has been developing this from the start. Targetgene is a pioneer in gene editing. We have 11 granted patents today and many different patents pending as well. The main problem that we've solved of gene editing is safety. Gene editing, of course, can be used to cut and paste the genes in the genome to cure many different heritable diseases or cancer or immune disorders. But the problem is how to cut only once in the DNA. And when you cut in the wrong gene, this is called off-target. So, and this is a major problem of CRISPR specific. The way we've solved the problem is through rational protein design. We actually designed a new platform, the TG platform again, which uses, instead of one guide, it uses 2 RNA guides to bring the nuclease which cuts the DNA to the right place in the genome. And the system only works when the 2 guides are at the correct distance from each other. That is, unlike CRISPR, where you have one guide, in the TG system, you have 2. So how does this work? You express in the cell, or you deliver to the cell, a protein and 2 guide RNAs. They self-assemble or they can be pre-assembled. The genome, which is 3 billion bases long, gets searched using the RNA guides, which hybridize to the target site at the correct distance from each other. The 2 proteins then dimerize, and like the 2 halves of a scissors, only when the 2 halves are at the right distance from each other do you get cleavage or cutting of the genomic DNA. When you cut a gene, you can either insert into this DNA your payload, be it a CAR, for example, or a checkpoint inhibiting molecule, or any other type of editing that you can imagine, or you can just knock out a gene, or you can replace a gene. So if we compare side by side CRISPR and TG, how many off targets you would expect, you will see in red on the left the number of off targets expected for each of these. Here we have the famous checkpoint proteins, PD-1 and CTLA-4. You have the T-cell receptors, for example, and there's another gene here, myeloperoxidase. So all of these you can see with CRISPR are expected to have a lot of off-target, and this is for the best guide you could choose for them. With TG, you will get 0 off-target for these targets. And not only are we getting off-target, but if we look at what those off-targets are, for example, for the first gene here, 13 of these are potentially involved in oncogenesis. So this is a huge problem for CRISPR. It's been recognized widely as the major problem of gene editing. So here's an experiment showing in primary human T cells, TG knocking out the gene, the checkpoint gene PD-1 in this case. So you can see we get highly efficient editing. The system is a very versatile one, where it can be delivered either by messenger RNA, or by protein, or by plasmid, or by virus. All these systems are equally viable. Of course, viruses are only needed if you want to do in vivo. We're talking today about ex vivo mainly, where you deliver either an RNP, that is the protein and the RNA together, into the T cells, for example, or the mRNA and the synthetic guide into the T cells, and then the TG will cut the gene and it will be edited, and then you can expand these cells and bring them back into the patient from which they were collected for autologous, or in allogeneic settings, then into another, from a healthy donor to a patient. So to sum it up, the TG platform, we have a proven IP, we have best-in-class genome editing platform, we have proven capabilities. TG will plug into the whole field of regenerative medicine, add safety and efficacy to products already in development, making them safer. For example, knocking out TCR in a car product, for example, or enable creation of completely new non-existent products today, which we can develop, co-develop. We will use Targetgene to disrupt a multi-billion-dollar market of novel indications and of existing indications. We're talking about one novel indication is pulmonary arterial hypertension, for example. This is together with another Israeli startup called Lempo Therapeutics, where we expect to reach a multi-billion-dollar market in something like 7 years. All goes well, but we need major investments to make this happen. For solid tumors, we're talking about the T cells mainly. This is, of course, a $500 billion market or so, and this is a bit of a longer development path, but there are many potential partners. We just need to find the best ones. And these can be funded either through these collaborations or by non-dilutive funding, And the last possibility is also ad hoc solutions specific to specific companies that need specific solutions, where we can talk about out-licensing and where we will need upfront funding to make those happen. So a bit about the team. We're a team of scientists. Dan and myself are the co-founders and managers of the company. We have with us a senior immunologist from the famous lab of Zelig Ishchar, the father of CAR-T. We have an oncologist and a gene therapy expert on our advisory board. We're doing COVID development with Lempo Therapeutics. We have collaborations in cancer with North Carolina and hospitals and universities here in Israel as well. Targetgene has had some investments from Adama, Bayer, and undisclosed Swiss CDMO. We've also had some funding from the Israel Innovation Authority. And that's it for my presentation. I hope I've managed to stay within the time limits. So somebody here has asked me about Caribou Biosciences. That's correct. Caribou is
Dr. Shiboleth, and we have a limitation of time with each of company. So please make answer on Q&A, not having an answer lively. So I'm just so I need to turn the session to the next company. Is it okay?
Okay, so I will answer in the chat.
Yeah,
Okay.
please.
Thank you.
Okay, thank you for your explanation. Yeah.
Thank you.
And then the next turn is the Korean company Genecast. The technical director Kyuwon Baek is going to have the pitch for the session.
I'm Kyuwon Baek, Technical Director in Genecast. And can I start? Okay. I'm Kyuwon Baek, Technical Director in Genecast. I'm glad to have a chance to introduce my company and technology. Genecast is a molecular diagnostic company, mainly focuses on liquid biopsy-based cancer diagnostics by detecting circulating tumor DNA in your body fluid, like blood, saliva, and urine. This table is a brief summary of our company. We have developed a new technology called ADPS, Early Discriminating Priming System. It's a real-time PCR-based.
네, 이 화면인데 여기를 읽고 계시는 거예요?
Sorry? 우리가 넘기는 게 아닌가?
저희가.
Okay, I'll start again. This table is a brief summary of our company. We have developed a new technology called the ADPS, Early Discriminating Priming System. It's a real-time PCR-based gene detection system, and I'm going to explain the detail of ADPS when it comes over later from the technology part. And we have several products based on ADPS, and they are CE certified or research use only state. Genecast is a research-oriented company, and most of the members are engaged in R&D and quality management. Next page. As I mentioned earlier, Genecast is a liquid biopsy-based cancer diagnostic company, and liquid biopsy is a technique that are using molecular biomarker in your blood. When comparing to tissue biopsy, it is more safe and convenient because it doesn't need any surgery. As you know, cancer treatment is the second biggest market after metabolic disease. Recently, liquid biopsy is one of the hardest issues in the cancer diagnosis. However, despite its numerous advantages, liquid biopsy couldn't have replaced the tissue biopsy market because its technical accuracy is still not enough. It's very complicated to find a tiny amount of cancer DNA from your blood. This figure shows the required detection sensitivity for accurate diagnostic based on cancer stage. Detection of early stage cancer requires a sensitivity of 1 to 0.01%. Here, 1% means that more than 1% cancer gene should be present to be detected. And residual cancer after surgery require even higher detection sensitivity. But the current state of art technology couldn't achieve the sensitivity of below 0.1%, which means early stage cancer screening or residual cancer monitoring has a kind of a technical barrier. Next page. To overcome the detection sensitivity problem, Genecast has developed a new technology called ADPS, which has obtained unparalleled high sensitivity of 0.0001%. It means you can find one copy of mutant DNA from 1 millionth copy of wild-type DNA. ADPS could provide a significantly high signal-to-noise ratio because it has a selectively amplified only mutant DNA without touching wild-type. Here shows several representative technology platforms in the market and their detection range. The bottommost arrow shows the required sensitivity for a successful liquid biopsy. And you can see only ADPS can cover the full range. This page explains the advantages when the higher detection sensitivity is applied to companion diagnostics. The table shows the reactivity of each cancer drug for each EGFR mutation. Actually, EGFR mutation represents lung cancer. If we could define the correct accurate genetic information from early stage, it would greatly improve the success rate of cancer treatment. Next page. It's another opportunity with a higher detection sensitivity when the pharmaceutical company conduct a clinical trial, and if they could pre-screen the cohort related to their drug candidate, they could save the cost and time and could raise the success rate of a clinical trial. From here, I explain our technology. Yeah, the figure shows the comparison of a real-time qPCR results. When you are using the conventional system, the wild-type gene, the blue color line, are amplified as well in addition to mutant gene, and it lowered the signal-to-noise ratio and lowered the detection sensitivity. On the right side, our ADPS system amplifies only mutant DNA very selectively, so there is no false positive problem, and we can maximize the detection sensitivity. Then how is it possible? To minimize the wild-type amplification, Genecast has developed a novel DNA polymerase. it has a remarkable discrimination power, so we can amplify only mutant genes that we want to find. Next page. This page shows how our new enzymes specifically amplify the target gene. ADPase enzyme has a single nucleotide discrimination power, so only primer with a perfect match with the template can be amplified. ADPase is not just a theoretical possibility. We have a CE certified and RUO product and following pipeline. Next page. And we have built a patent portfolio and currently 3 approved patent and 19 progressing from abroad, including US, Europe, EU, and other major countries. If I recap today's talk, we have developed a new technology called ADPase to overcome the technological problem of current liquid biopsy market. And it has a highest detection sensitivity, and it has a big potential that reorganize the market from tissue biopsy to liquid biopsy. Thank you for listening. It's my talk. Yep.
Thank you, Professor Dr. Baek. Let me wait for a second. We have there is some minutes left, so if you have any question, please raise the one. Otherwise, it can be passed to the next session of AlphaTAU. In the meantime, if you have any question, just using the Q&A button on the bottom side of the screen. And there's Mr. Ronen Segal, the CTO of AlphaTAU, will gonna be the pitch at this time. Ronen, the floor is yours.
Oh, thank you very much. It's a pleasure being here, and I want to congratulate all the colleagues that are speaking, really exciting technologies. So I'm going to tell you about the AlphaTAU company, and our technology is called AlphaDart. And I believe that this is the Bio-Convergence in its best, where in our development, we are combining biologists, physics, chemistry, all sorts of engineering to bring a new hope for cancer patients. And we're doing it through a new modality of radiation therapy. Basically, it's taking the most deadliest nuclear energy and turning it to the most novel cause of treating cancer. In a nutshell, our achievements today is that we first time in healthcare are able to harness alpha particles to be able to treat solid tumors, and I'll explain how we do that. And we've already done several clinical studies which validated the safety and efficacy of the technology and had a very impressive responses, almost 80% complete response of treatment of cancer tumors. We're conducting right now multiple clinical trials in leading places in the world. And we're looking forward also for finding collaborators in Korea, both for clinical studies, for research, and also for distributing in the market. The FDA has acknowledged our technology as a breakthrough device designation for 2 indications already, for superficial tumors and also for GBM, for brain cancer. We have a very strong family of patents, and we're supported by leading investors. So now, just in a nutshell, to put everybody in line, when we're talking about a radiation therapy in oncology, usually we're talking about gamma radiation, which is high energy photons, or beta radiation, which is high energy electrons. And what we are using first time is alpha particles, which are heavy particles. They are very, very potent. They have much more energy than any other particle, but because they're heavy, it's very hard to make them move inside a biological tissue. So practically, as you can see here in the drawing, an alpha particle, if I take a sheet of a paper, it will be stopped, which means that it's a very localized type of treatment, whereas beta or gamma will very easily penetrate any type of material, and we need hard concrete or at least aluminum to be able to stop it. Now, the big benefit of the alphas versus the existing radiation therapy is that when it hits the DNA of the cancer cell, it causes not only a single strand break like the old type of radiations, but it causes a double strand break or even a cluster strand break, which the DNA could not repair from that. Moreover, the alpha radiation does not need any oxygen, which means that also hypoxic tumors, which would be resistant to regular type of radiation, would not be able to resist the alpha radiation. And also the localized means that we have a very safe type of treatment. Now, as I said, the beta and the gamma, when you try to treat a certain area of a tumor, as you can see here, it will radiate also the surrounding tissue, and unfortunately will cause a lot of collateral damage and will damage also healthy tissues. Whereas the alpha radiations, wherever you put it, you just place it inside the tumor, and we'll see just in a second how we do it, and it stays there. It doesn't go anywhere, so you don't get any side effects. Now, this is a demo how it actually works. So what we're using is an applicator with a loaded seed of 1 centimeter needle, which is loaded by radium-224, which is our emitter of the alphas. And it works like a cluster bomb. When it goes into a tumor, as you can see here, we release the seed inside, and then all the coated isotopes that are on the seed are starting to be, by recoil force, recoiled into the tissue. And then we get a much larger destruction zone, which at the end, we get a whole decay chain of about 5 millimeters in diameter of destruction in the tissue. And this is the main breakthrough of AlphaTAU, which enables to get a cloud of destruction inside the tumor, which is not enabled with alpha particles before. And once we have this, we can treat a whole volume of the tumor by placing several seeds like that. Now, the way that we're getting it is, as you can see here, the coverage of each seed is going to be about 5 millimeters. And the good news is that in healthy tissue, if we take this and put it in the healthy tissue, the diffusion is much, much shorter, so we cannot cause any damage. And this is mostly because of the chaotic blood configuration that we have in tumors. Now, the way to be able to cover larger tumors is by treatment planning according to a CT that we have of the patient of the tumor, and then we can place these seeds in spacing of 5 millimeters, making sure that we're covering the whole tumor. And as you can see here, we could be very close to blood vessels, where usually these type of tumors would not be treated with radiation because we're afraid that will cause damage. With alpha, we have no problem to treat this. So we started with many solid tumors, animal models, to see which one will be resistant to the alpha. And we haven't found any type of cancer which was resistant to the alpha radiation, which led us also to look at the influence of the immune system. And what was also nice is that we have not only a local treatment, but also a systemic effect, which synergizes with the effects of the immune system. And here we can see that not only we could generate an immune effect where mice are re-inoculated with the same tumor, but it's also specific to the type of cancer that we're treating. Now, just to show you a few showcases of patients that were treated. So this is, for example, an 8-year-old where he had 2 cancer lesions on his ear, and the only option was to cut off his ear because he already got radiation and it was radiation resistant. And here we placed those seeds, and you can see that after a month, his ear is intact and there is no signs for the cancer. Another case is a scalp, and here you can see that after the treatment, you can't even notice where the cancer was, and this patient is totally clear. Another one is more complicated cases like tongue. Instead of cutting the tongue, we are able to save the tongue and to make these patients healthy. So all in all, we're treated today more than 100 patients, mostly in Israel and in Italy. Now we finished a feasibility study in the US, and we are going into our pivotal multi-center study for getting our first commercial approval for superficial tumors. And as you can see here, the results are phenomenal. We got 80% complete response. All the rest of the 20% were partial response, which means that we had 100% response to this treatment. Not only that it's very effective, it's also very safe, and we showed that the toxicity is very minimal. We are now actively working in many countries and prominent hospitals around the world, as you can see here. And we started working also in Asia, in Japan. We have several sites, and we're looking forward to extend this with additional partners. Here, what's nice in this picture is that you can see the operation. If you're used to radiation, usually you need shielding. The staff needs to wear special protections. And here you can see that with the alpha, because it's so short distance, no shielding is needed, and the operation is very simple. It's actually being done on a dental chair, and you can see that all the staff here are very casual. I'll skip this just to show something really phenomenal. We have a patient where it was a lady. She had cancer on both of her legs, and we decided to treat her first leg in the first visit. But we were very surprised that when she came to the second visit to treat the other leg, the other tumor went away without any treatment. And here we see a clinical evidence of the systemic activation of the alphas that are treating not only locally, but also elevating the systemic effect and being able to treat also possibly metastasis or other tumors in the body. I'll skip this just for sake of time, but I'll show you that one of the challenges that we have is that we're looking also for partners for our logistics and production, because we're talking about isotopes with fairly short half-life of about 3.6 days, which means that we need to be close to the hospitals. So right now we have production facilities in Israel and Tel Aviv and Jerusalem. We have one in Massachusetts and another one in Japan. And these ones will cover the regions, but down the road, we'll see that we would need to open also logistics operations and productions in any country that we'll be working in. I think I'll skip this, but I'll just show you that the next challenge that we have is how we deliver these seeds for different organs. So here, for instance, we have a collaboration on developing a penetration of these seeds into GBM patients with brain tumors. And there is a very, very delicate way to deliver these seeds in a safe manner. We have a very experienced executive team, again, Bio-Convergence. So all our executives are coming from different disciplines working together, very strong advisory board and board of directors, which comes with a lot of experience from global companies. And altogether, we have many partners, and we're looking also for partners in Korea to take this and to save as many lives as possible in the world. Thank you very much.
Thank you, Ronen. There is one question to you on a Q&A the function, so please make an answer on that. And the next session is a pitch from the BioPharma. Mr. Yeongsun, the CTO of BioPharma, will give a pitch. So the floor is yours, Mr. Choi. Thank you.
Hi, it's my great honor to introduce our company, BioPharma, to all of you today. Our company is quite new. Actually, we are established about 2 years ago. Let me briefly overview our company. We established 2 years ago and working on various pharmaceutical drugs and novel materials. And our company's main focus is on the drug delivery system for the various indications such as cancer and also the Alzheimer's and COVID-19. Our company has the identity as a Bio-Convergence technology company focusing on the drug delivery systems. Our delivery system has various functions such as cell penetration and blood-brain barrier penetrations, and also the skin penetrations. So our pipelines consist of 3 indications and 1 platform technology. As you can see here, we have 4 pipelines here total, and we are focusing on the solid tumor for especially the colorectal cancer. We are now at the end of 2021 and working on for the phase 1 IND with the various tox and CMC studies. In the next indications, we are working on the neurodegenerative disease such as Alzheimer's disease. And this work is also in the preclinical stages. And our major focus these days are now more exerted on the mRNA vaccines using our drug delivery platform, as well as the novel small molecule therapeutics for COVID-19. This COVID-19 vaccine technology is quite new. However, because of these various variant emergence these days, we are now also working on the Delta variant mRNA vaccines as well. Actually, as I said, as I mentioned earlier, our major focus is on the DDS platform technology. We are here that the SC6 is our proprietary materials for various delivery functions. So here, let me briefly introduce our drug delivery platform. We call it as a BP401. This DDS platforms has quite been in the market quite a long time. However, because of the emergence of COVID-19, this delivery platform has gained a lot of interest for worldwide, as you can see from the Pfizer and Moderna vaccines, in which they use the lipid nanoparticles. In our technology, we use the sorbitol-based materials, which is quite safe from the perspective of FDA safety guideline. And also we introduce our delivery functions. We added the guanidine unit, which is one of the small unit of the arginine unit, so that it can bind to the negatively charge the membrane of the cells or skins or tissues. This technology has been developed from in the previous page, Dr. Tae-Jong, who was the president of the POSTEC in Korea, and has been published in the various SCI journals for their intracellular penetrations and BBB penetrations, and recently transdermal skin penetration. This is the summary of their papers, and I will skip this. And for this technology, one nice thing about this SC6 is that it can allow for the molecules to pass through the BBB with a high specificity and high penetrability. And also in terms of the penetration to the skin, with the drug delivery molecules, it can deliver the large sized proteins, also antibodies across the skins. In our major, one of our pipelines is also focusing on the cancer, especially for cancer stem cells. This material is quite new. However, this structure is based on the artemisinin, which is quite well known for their activity against malaria. We are working together with our Korean university study group, to publish 4 patents until now. And as you can see, the cancer stem cells are quite important to prevent the relapse of tumors or regression or relapse of tumors, as well as the movement of tumors as well. So we are focusing on the both cancer cells and cancer stem cells for tumor growth inhibition. The artemisinin-based drugs are known to cause cancer cell death by ROS generations in the process of binding to iron ions, which is present in quite large amount in the cancer stem cells as well as the cancer cells. So which called as ferroptosis. So our molecule has very specific endoperoxide structures. So when it enters the cancer cells or cancer stem cells, it binds to iron, it produces the ROS, that it causes ferroptosis. In our in vitro study has shown a high specificity toward the cancer stem cells as well as cancer cells in both in this cytotoxic assay and sphere formation assay. In this preliminary in vivo efficacy study, we have quite a pretty preliminary, but we have some significant decrease of the tumor growth compared to the negative control. And in addition, we are working on the anti-dementia therapeutics. This compound is a single compound, which is originated from the Japanese ume fruit. So we have been working together with the kids in Korea to find out how we can develop this ume flower, which is known to in the literatures to be developed as a novel anti-Alzheimer drug. So for this efficacy of these ume flowers, we have 2 different papers to show the in vivo efficacy and as well as the oral toxicity. In a third pipeline, we're working on the mRNA vaccine using our own proprietary S6 platforms. As you are all aware of, the Pfizer, BioNTech, and Moderna vaccines have high efficacy. However, there are problems in their storage and their the thermal stability. So we need the novel drug delivery platform to enhance their thermal stability as well as their storage issues. As you can see here, the lipid nanoparticle is very nice structures. However, in terms of the production, it has quite a lot of issues, and also as well as the safety issues as well. So what's unique about the BioPharma's mRNA vaccines are their fourfold. One is our drug delivery platforms can allow us to freeze dry the vaccines, so you can easily distribute around the tropical regions or other underdeveloped countries. And also in terms of the scale of production issues, we use the liposome technology, which is quite available in the around the countries, around the world, so we can easily scale up the production of these ribosomes. And the unique things about our platform is that we convert the S6 platform to the riposome so that we can make it as our proprietary drug delivery system, so you can evade any kind of patent issues. This is the simple scheme of our simple step of the making mRNA vaccines. Until now, we have successfully made the lyophilized injection formulation with the good stability at room temperatures. And in our mouse studies, you found that the good neutralizing antibody generation in the mouse studies, in either liquid form or lyophilized form. Another indications we are now currently working on is the COVID-19 therapeutic. So for the game changer of this COVID pandemic, we need to have some kind of therapeutics, which should be orally administered. So until now, the two directions have been pursued in a worldwide to make to develop our COVID-19 therapeutics. One is drug repositioning, and second is the development of new substance or formulations. As I mentioned, we have been developing new small molecule therapeutics for the COVID-19 therapeutic. Until now, we have screened about 50 small molecules to find out the 4 hit compound with this similar activity to the remdesivir in vitro anti-virus screening test. And we have performed the in vitro test in our Korean company, a Korean funded Institute Pasteur Korea here in Korea, and we have patented this. We applied the patent applications early in this year. Basically, our business strategy is threefold. We'd like to maximize our business performance by utilizing our platform technology, as well as independently licensing out each pipeline. And our strategic portfolio has been focused on very well known indications, such as solid tumors and dementia and COVID-19. As you know, this is the largest share in the pharmaceutical market. So with this, our endeavor and our success in the various indications, we are hoping that our nano-bio-convergence technologies allow us to pursue these indications to be able to get into the market in the future. Thank you for your attention.
Thank you. Thank you for the nice pitch. Next session is the C2I Genomics from Israeli side. The Boris Oklander, the co-founder and the CTO, will give a pitch. So Boris, the floor is yours.
Thank you. I'll share the screen. Okay, do you see the slides?
Yes, perfect.
Okay, great, thank you. So C2I Genomics operates in the field of cancer intelligence, very close to what was presented by Genecast in the previous session, but we take a different approach. So we basically, again, what we are doing, we are diagnosing and monitoring the state of cancer patient by a simple blood test, and we use AI and the whole genome sequencing to really detect the recurrence of the cancer very early as compared to any other available technology. We are a relatively young company, but with great progress. We raised more than $100 million from the inception. We're addressing a really high need and a multi-billion-dollar opportunity. We have a presence in different locations in the United States and in Israel. We have developed a leading clinical platform. We'll talk about that, which allows in a distributed manner to engage with different pharma companies or even in diagnostic space, patents, defending our technology. And we have already established a global distribution network in different regions in the world. We see ourselves as a cancer intelligence platform that enables different kind of applications. So we generate a lot of genomic data to provide one-stop shop for the oncology, for biomarker discovery, And we provide a lot of analytics and advanced AI methods to support all these breakthroughs. I think the main competitive advantages are the rapid turnaround, which are important for clinicians to make decisions in landmark points when they need to decide on treatment with high sensitivity, high specificity, and also what's important here as compared to other technologies is low input requirements. So this type of solution addresses different markets for the biopharma. Then there is a large segment of recurrence monitoring, because today people that are going through surgery to remove cancer use basically are for monitoring them using imaging techniques that reveal the recurrence of the cancer really at late stages. We can guide and make decision making for the therapeutic selection, and also we can repurpose our technology for screening of the population for cancer. The idea is that we provide one test that can basically support the entire patient journey in the oncology. So we're talking about even before surgery, the new concept of organ preservation when the patients respond to neoadjuvant therapy, and our blood test can detect the complete response. So we remove the need of the surgery in many places. We've shown this in bladder cancer and rectal cancer. Then we can, by measuring the minimal residual disease, to inform the treatment and enrollment in clinical trials, and we show high performance for colon, lung, and bladder patients. And also we participate in clinical trials to support the immunotherapy treatments and monitor their efficacy. The idea is that basically about the technology is that we are patient specific, so it's a personalized medicine approach. We take the biopsy from the patient, so we have normal tissue and tumor tissue, and we derive from the both the mutation signature, which is personalized, and it's across the entire genome, which comprises thousands of point mutations, copy number variations, etc., which are very individual. And we have like a fingerprint for the patient cancer. Then during the treatment and during the patient journey, we basically collect blood tests, simple blood tests, and by comparing the genomic content of the circulating DNA in the blood and comparing it to the signature of the patient, we can basically detect the tumor burden, and we report these results to the physicians with very high accuracy. So this is basically the main idea about the technology, and the innovation here is, as opposed to the prevalent approach of using panels for targeted panels, or even tailored for customer, which are looking for, I don't know, 16 probes or 500 probes, etc. What we do shallow whole genome sequencing. So we're looking at the entire genome, and we have more opportunities to detect these mutations that were found previously in the patient's fingerprint, and therefore we increase significantly the probability of detection, even in really with low coverage and even in very low tumor fractions. So here you can see some representative results. So again, the test provides really high sensitivity with high specificity. We have shown reproducibility by comparing the same samples were sent to different labs in the world where sequenced, and here you can see the high correlation between the results that were blindly processed at different labs. This is the lab in the United States, and this is in Europe. we see very high concordance of the test. One of the additional things the high sensitivity allows us is really early detection of cancer recurrence. So you can see here a few examples of patients that had a recurrence. This is the time axis, and the clinical imaging really detected the cancer recurrence late as compared to our test that can basically detect the cancer recurrence within weeks after the surgery of cancer removal. Our platform generates a lot of data. So from all the clinical collaborations, we are projected to have a lot of whole genome sequences coming from real patients around the world with a lot of different indications. This database support different applications for tests regarding, as I mentioned before, the organ preservation, immunotherapy, and monitoring of cancer recurrence post-surgery. Again, our offering to the pharma includes enrollment of the patient to clinical trials so that after the surgery, we can detect whether the patients are with high risk of cancer recurrence or not. And this might be the criteria to including enrolling the patients in the studies, because other options are really poor. Then based on our test, we can monitor what happens to the patient, how they respond to the treatment. And then our test also provides a surrogate marker for complete response. So we don't need to wait for years for the survival analysis. After the completion of the treatment, we by consequent tests, we can know whether the treatment was efficient or not. On the other aspect of the technology, other than test, we developed a very flexible cloud platform that basically any lab in the world that has a whole genome capability, so basically a Illumina machine or something, a sequencer, can basically using a commercial off-the-shelf kits do the sequencing at their local lab and just upload the data and send back the report. The platform itself is compliant, so GDPR compliant and HIPAA compliant, and it is very flexible to repurpose. And in different collaborations, we can adapt really what we do there to accommodate different questions, different volumes of data, genomic data. Just to give you the idea, one blood sample is translated into a digital data volume of more than 100 gigabytes. So with this approach, C2I Genomic is able to engage with thousands of sequencers that are already deployed in the world. And this is why we are looking also in the collaborations around the world to provide this capability. The summary of the advantages of this technology is we require only very little input material. So 1-2 ml of plasma is enough. Using AI, we can provide ultra-sensitive results because everybody knows that basically the genomic data that is sequenced is noisy data with a lot of variability, and we came up with technology that can suppress the noise very efficiently. We do really personalized medicine. We look into genomic makeup of the patient and his tumor to track really with high sensitivity and specificity a minimal residual disease at really low levels. Additional thing, because we look at the entire genome and we work with all these large volumes of data, we are able to incorporate in our analysis the various somatic mutations, whether these are SNVs, CNVs, and others. Again, our approach, as opposed to centralized lab, we work in the distributed manner with many labs. So we have a global processing network, and we collect a lot of data that basically can, by data mining, can guide us to the promise of personalized treatment nomination. So we have really extensive team, executive team of people that were working in different industries around the world with a track record of success in both the genomics, pharmacology domain, reimbursement, et cetera, and really high profile advisory board. And in this collaboration, we are looking for basically to distribute our technology in Korea. This is one, and to collaborate with other companies, mainly in the therapeutics domain, where our solution can provide the monitoring and the evidence of the treatment that they provide. Okay, so thank you.
Okay, thank you, Boris. Thank you. And the last company is from Korean side, the KBioLab, and the CEO, Mr. Tae-Jong Kim, will give a pitch. So, Mr. Kim, the floor is yours.
Thank you very much. Let me start. I believe it's ready. Okay. So I will start my presentation. My name is, thank you. My name is TJ, the founder and CEO of KBioLab. Thank you for giving me a chance to present my company KBioLab's technology. I will mainly introduce the treatment of atopic dermatitis using skin microbiota improvement and briefly introduce various technology my company has too. There are many atopic treatments around us, and various products are sold by other companies. However, many people are still suffering from the atopic dermatitis. The medicine for the atopic dermatitis are hormones such as corticosteroid or antihistamines. But because of side effects, they cannot be used regularly. There is no effective treatment for atopic dermatitis for long-time use in every day. This suggests that a new approach is needed in the treatment of atopic dermatitis. It is known that the cause of atopic dermatitis are genetic factors and environmental factors. Recently, skin microbe has been suggested as one of the environmental factors. The figure below the slide shows the destruction of the skin barrier at the site of atopic dermatitis by Staphylococcus aureus, a skin pathogenic microorganism. Many Staphylococcus aureus exist in the area with atopic symptoms, and many scientists report that removing the Staphylococcus aureus improving the atopic symptom. In order to improve the symptom of atopic dermatitis, antibiotics and bedding with diluted bleach are used to remove Staphylococcus aureus from the skin. These methods are used medically and known to be very effective. Even they are not good for the skin itself. Therefore, we need a safe and effective method of removing Staphylococcus aureus that can be replaced current unsafe, slightly harmful medical method. What you see in this slide is there is a demand. For the atopic treatment that remove the microorganism in a new way in the scientific journal related to the skin. KBioLab has developed a commercialized product and meet this demand. My company KBioLab discovered a compound that improved the skin microbiota from over the 400 edible plant extract and developed the VSP complex to maximize the skin microbiota improvement. In the current market, the method of directly using living beneficial bacteria or using the extracted compound from the beneficial bacteria was nothing to do with change in the skin microbiota. KBioLab has a true skin microbiome technology that uses plant extract complex to improve the health of each individual's unique skin microbiota. Clinical tests show the improvement of the skin microbiota after 2 weeks use. Staphylococcus aureus, a causative agent of atopic dermatitis, decreased by an average of 29% in the skin of 21 people. And Cutibacterium acne, an acne bacterium, decreased by the average of 23%. These clinical tests of improving the skin microbiota are known to be the world's first and unique result obtained by the KBioLab. Various positive skin health effects also shown as the skin microbiota improve. All of these improvements are what we can expect from the VSP complex to improve the skin microbiota. This is how the customer face is changed by KBioLab's product after 1 year use. The redness of the face back to the normal. We tested the microbiota change on the face too. As you can see, the yellow color on the plate with Staphylococcus aureus was switched into the red color on the plate with Staphylococcus epidermidis, which is a beneficial microbe on the skin. When applied to the patient, the treatment result was superior of those of medical ointment. This slide shows only some representative result among many consumer results. In particularly in the case of infant under 1 year old, it can be used safely without any side effect. And the treatment was very effective, resulting in the return to the normal skin. This slide shows the product of KBioLab using our technology. The US market, the number of patients with atopic dermatitis requiring treatment is 9.6 million, and the market size is $3.8 billion US, considering the annual consumer cost of the product. The average annual market growing rate is 13.4%. KBioLab has a library of the more than 400 edible plant methanol extract and 80 their constitutive compound. In addition to the material and technology introduced in the following slide, we are looking for the partner to develop the various compound and medicine using this library. Our first market using the skin microbiota improvement is atopic dermatitis. However, KBioLab's VSP complex is also effective for acne and removing body smell. In the clinical test that showed the previous slide, VSP complex effectively removed the causative bacteria of acne and control the sebum secretion. And improving the skin microbiota will remove the body smell from the beginning. Removing the body smell by improving skin microbiota is a new and innovative method and it is the only way to eliminate the smell from the beginning. We have discovered a compound that improving the intestinal microbiota by using the same technology to improve the skin microbiota using plant extract. As another technology, we are developing the skin diagonal technology. The microbiota of the skin is mostly affected by pH. We are developing a patch that easily measures the pH of the skin. By fusion of this technology and antibody technology, we can be used to selectively and quantitatively measure the microbial species of the skin. It can be also applied to the development of the sensor for measuring the skin cancer. We are also developing next generation antibiotics that kill the superbacteria without emerging resistant cell. This compound inhibits only virulent specter of the pathogenic bacteria. We have developed that suppress the number of first cell when the antibiotic kill the bacteria. There are some bacteria that cannot be killed because their metabolism is stopped. The compound of KBioLab reduces the number of first cell when antibiotics are applied. This will be very effective in the treatment of tuberculosis. The compound that inactivates Staphylococcus aureus and Candida albicans, and then coronavirus, including Delta mutation, has been discovered from natural extract. Hand sanitizer, such as ethanol, damage the skin. The sanitizing agent that does not damage the skin is essential for continuous hygiene in the ongoing corona pandemic situation. As a startup company, KBioLab is looking for the partner at any level of the business. Thank you for your time.
Okay, thank you, Mr. Kim. This is the last company of our company session. Then there's going to be the panel discussion of IIA and the KORIL-RDF. Before starting the panel discussion, I just let you know that there's going to be the 4 windows going to be pop up at the end of the panel discussion. So please make an answer. It's a very short one. So please bear in mind that one. And I will pass this mic to Ella Heller as a moderator for this panel discussion. So Ella, please make a start.
Thank you very much. First, I would like to say thanks to all the companies and all the innovative technologies. I think it's just a sample of what's going on here in Israel and also in Korea. So it's just a glance of what's happening, and we are more than happy to have the chance to show you the innovative technologies in bioconvergence. I would like now to invite Seongryong Kang, the Chief Executive of KORIL-RDF, to show what the joint fund between Israel and Korea is now doing in the regard of bioconvergence. CE, please, the stage is yours.
Could you see my slide full screen?
Yes, but we see also the Word file.
Okay, how's it going? Okay, good afternoon and good morning, ladies and gentlemen. I'm Seongryong Kang, Chief Executive of KORIL-RDF, which stands for Korea Israel Industrial R&D Foundation. And it's my great pleasure to have and join this webinar with outstanding bio companies from Korea and Israel in cooperation with Israel Economy and Trade Office Korea and Israel Innovation Authority. Well, KORIL is a binational foundation funded by the Korean government and the Israeli government. The mission of the KORIL is to support bilateral technology cooperation between Korean companies and the Israeli companies to bring the economic impact of both countries. For this, KORIL has served as the single window for wide cooperation between two countries to promote collaborative industrial R&D, support joint venture for commercialization, and facilitate technology transfer between two countries. As you know, following COVID-19, Korea and Israel proved themselves a high level of capabilities in the field of biotechnology and healthcare technology, and both sides have substantial potential in so-called bioconvergence between biotechnology and the other various technologies such as nanomaterials, AI, big data, IoT, and so on. And last year, KORIL announced the Bio-Convergence initiative. Also, with the amendment of the bilateral agreement for technology cooperation between the Korean government and the Israeli government, the KORIL has launched KORIL Strategic Program to support big R&D projects in strategically important sectors. and the first RFP of the strategic program focused on bioconvergence. You can see the critical timeline and data in this slide. The call for proposal is already opened from 13th October this year, and the application close date will be by 10th February next year. Well, this strategic program will follow the same rule and procedures as the KORIL general program, with a few exceptions. The grant is increased to 30% to 50% of the total eligible project cost, up to $3 million and will last 3 years. You know, KORIL only funds joint R&D project undertaken by private companies from Korea and Israel, and you know, academy participation will be highly recommended as a subcontractor. The budget for subcontractor is up to 30% of the total project budget, unlike the KORIL general program, that is just 20%. And subsectors for bioconvergence project proposal are in this slide. Please visit KORIL's website for submission details. And also, if you are already having a project idea and are looking for partners, or you are interested in participating a project proposal, given your expertise in the field, but haven't yet formed a project idea or proposal, then please visit KORIL's webpage, www.koril.org. Thank you for your joining today's event, and feel free to contact KORIL to discuss your proposal. Thank you for listening.
Thank you very much. We want to move to Karina Rubinstein, Senior Director in Business Development in the division of the Startup Division in the Israel Innovation Authority, to just share a bit of what's going on here in Israel regarding bioconvergence, and of course, to share what the Israel Innovation Authority is doing now. So please, Karina, the stage is yours.
Thank you, Ella. Good morning, everybody. Good afternoon. Can I share? Thank you. Okay. So just a second. Good. Okay, so just a few words about bioconvergence. You all heard of the previous presentations and companies from Israel and Korea. We are really happy about this progression in our collaboration through KORIL. It's exciting for all of us. The fact that we are opening, and the second time already, a call for proposals in this area is really not trivial. And we are really hoping to see very good projects coming in from both sides. I think both Korea and Israel are now at the same spot of understanding that the future of medicine is going through transition, and the fact is that we have a very good point in time, both from the worlds of engineering and biology, that are both colliding now to enable this kind of a multidisciplinary approach to solutions that are challenging, that are facing the challenges of the world in healthcare. So we saw, all of us, what is happening going on the COVID year, and the fact that we are not over the pandemic yet. And I think my next slide, which is not so updated, I think the timelines here are shorter than showed here. That was done, I think, a few months back. But now the fact is that we can identify new mutations in a fraction of a time. It's not 10 days. I think it's much lesser than that. And the fact that we can diagnose better than shorten time than 15 minutes, by all means, develop new vaccines in a staggering short time now. All of that is enabled, and you saw that through the presentations, the previous presentations today, by technologies that are coming in from the convergence world, and the engineering side is really dominating here. I think, you know, also this slide, we've been talking about it for almost, I mean, it's over 2 years now, about the integration between biology and engineering sides. You all saw the strength of the engineering side and all the projects that you saw before me. And I think it's already dominating so much this world. And needless to say here, the integration between the worlds of genetic engineering, which has been showed here, nanotechnology, AI, physics, and advanced materials, and other fields as well coming in from the engineering side. It has to be collide to the biology side. Both these worlds are really providing the true solutions that we all need in very short time. And we have to emphasize that. So we are talking here about a really huge opportunity going forward. The market would be $4 trillion, which the areas of human health are dominating this market by 45% of it. It's a huge opportunity for all of us. Both Korea and Israel can join forces together and tackle and tap into this huge opportunity, economic opportunity together, bringing to the world cutting edge technologies and true solutions. I think it has an implication in all the facets that you look in when you're looking at transformative impact that this area is providing the biopharma world, traditional as we know it, but it's going through a very massive transition now. It has automative lines now, automative industrial drug discovery and clinical trials and so on. This is transitioning the world into a more cost-effective drug development. You saw that before me in the previous presentations, and also more emphasis on more personalized therapeutic solutions, which we all need. I think that Israel has a lot to offer. You also saw that in the previous presentation. The AI scene here in Israel is really a powerhouse, coupled with all the high-tech industry here in Israel. We have a very good medical system with centralized medical data that can be tapped in for future research, and very strong commitment from the government. And you saw that with our engagement with KORIL for funding and co-funding in these areas. So just to showcase, I mean, beyond C221 that you saw here and AlphaTAU, there is an abundance of Israeli companies growing. This scene is really growing by the day here. All of them providing a kind of an holistic approach to how do we treat diseases, how do we provide early detection and diagnosis, and very personalized and precise treatment solutions. All of these companies have a strong engineering side in their approach to providing cutting edge solutions. So our vision, and we really would like to do that with Korea, is to promote a cutting edge cluster in the area of bioconvergence and develop a strong ecosystem together, both our countries, and be the leaders in providing the world the solution it really needs. I think our collaboration here is both in the research side, academia and industry together. And the opportunity is endless from both our sides. I think we both have very strong areas that we can complement each other and do very good activity in the near future. So thank you very much for the time. I'm hoping that this session will be successful, and by all means, the collaborations that we see in the projects that will be submitted within this open call will be what the world is looking for. So lots of good health, good success, and good health to everybody. Thank you.
Thank you very much. I think I will bring back the stage to Ji-hoon to say a few closing remarks. But thank you again for all the participants. And of course, regarding KORIL, if you have more questions, please feel free to reach out for the Korean office or to the Israeli office to get more information about the current open call in Bio-Convergence or further detail about collaboration in R&D between both countries.
Okay, thank you, Ella. Well, thank you, everyone, for the long time joining. This is the Bio-Convergence webinar, and I really hope there's some of a good progress between the